I wouldn't identify them unless they want to be identified. And it was a question on effectively nucleoside phosphorylases, okay, which was, there isn't one for adenine because there isn't one for adenine. And now the question, and when I went back and looked at the diagram and then went through the book, where does the free adenine come from, which is the substrate for adenine phosphoribosyl transferase? And I can't find it. I can't find where it is, okay, because adenine, AMP is broken down to adenosine, right? And then, and then adenosine is then transaminated. Deaminated by adenosine deaminase, which is the one that's important in the bubble boy deficiency disease, okay? So, and then it goes, and it becomes hypoxanthine, okay, or inosine, and then effectively it's treated as hypoxanthine, so in that way. So there's no, I can't find a way that free adenine is there, but there must be another pathway because we wouldn't have an adenine phosphoribosyl transferase, which needs free adenine. So maybe between now and Monday, somebody can find it. I've looked through the book and I've really not there, and I've been teaching from that diagram for probably 8 years, okay, and I didn't realize it, and I think it was a very good question in terms of that. So, but there's no other way because there's nothing that takes adenine off of AMP, in terms of the way it works for the others. Okay, so today and these two lectures are relatively short. Together they're only about 17 pages, okay, so they're relatively short. There are some things that are going to be repeated. So for instance, arginine synthesis occurs in the what? Urea cycle, okay? So effectively, that's to redundancy, okay? So we should be able to go through it reasonably well, okay? So I'd be happy to answer questions and things so that I think we're going to have enough time in the two lectures. And even with taking out, I think I'm ending at 2:35, okay, or something like that, or it'd probably be before. But I'd be happy to answer questions, okay? So next week is I have 2 lectures and then review on Friday, and then I'm gone. See another class, okay? So in January or February. So, okay. So this lecture is going to be talking about amino acid anabolism, okay, which is the synthesis of amino acids. So which group of amino acids are we going to synthesize? The non-essentials, okay, all right. And how many non-essentials are there? Well, how many, yeah? It's 10 or 11, depending on how you look at it. You have to decide whether arginine is in Pirate Tim Hall is how many letters? It's 10 letters, right? But if you pull arginine out, okay, then it's only 9, and so you actually make it 11. What's the 11th one? Do you know? What? No, not cysteine. The 11th one, only it would become an essential amino acid if we had a deficiency of PLP or cystathionine synthase or cystathionase. Okay, so that would be the only time it would become essential. But for instance, yeah, tyrosine, yeah, tyrosine is the one because it's made from what? Phenylalanine, which is the right, which is an essential amino acid, right? So, okay, so tyrosine is an essential. It's the T in private, okay. So it's tyrosine. So no, I mean phenylalanine is the P in private, okay. That's what it is. Very good. So why don't you just stop me, okay, when I say something? You know, all of a sudden, okay. All right, so it's the P. All right. So what we're going to do is now look at that. So we're really only going to be looking at the 11 amino acids, and we've actually looked at tyrosine pretty much already when we did phenylalanine, okay, breakdown, so effectively we've kind of touched it, okay? All right, so it's not working. Use your magic, okay. Okay.
It should work for you now.
Okay. Okay. So I should have the diagram. Yeah, that's what I wanted. There. Okay. All right. So here we have kind of just a schematic of what we're looking at. The ones that are in yellow or beige or whatever that are the ones that we're actually going to be looking at the synthesis of them, okay, on that basis. The one that you want to add in here, because it's not one that's programmed for, it's an amino acid, but it's not coded for in protein, is ornithine, okay? And so we get ornithine from the breakdown of arginine to ornithine, okay, and urea, okay, and so we get ornithine, and then the pathway, this cycle continues. But there is a synthetic pathway, and glutamate semialdehyde. Does anybody know what a semialdehyde means? Instead of having two bonds, it only has one? That's not what it means. Okay? What it means is how many carboxyl groups does glutamate have? How many carboxyl groups? This is a tough one. The choice is 1 or 2. 2, right? Okay, good. There are 2, right? So what happens in those two, what happens is not the amino carboxyl group, but the other carboxyl group gets converted to an aldehyde. It gets reduced to an aldehyde. So one of the glutamic acid, where one of the carboxyl groups, the one that isn't the alpha carboxyl group, that one is in the aldehyde form, so it's considered glutamate semialdehyde. And it's going to be the way that you make proline and ornithine. And yeah, I think that's pretty much it. And so that's why. And then so it's the synthesis of arginine is the way that we're actually going to make it. Okay. So we see the relationships. There is a strong relationship in terms of making these to these pathways. Okay. So for instance, this is kind of here part of the Krebs cycle. Okay. Isocitrate, alpha-ketoglutarate to isocitrate to citrate, and effectively we can go in this direction, okay? So for instance, oxaloacetate can synthesize aspartate by what type of reaction? How can I make aspartate from OAA? How do I get most amino groups into a molecule? What process? Yeah? Yeah. Say, guys, come on. If you want the surgeon who's operating on you to whisper to one another, I think it's this, okay? And you know, you really got to be there. You got to let it, out there, okay? And if the good doctor can talk, okay, you guys can do it as well, okay? All right. That's one of my favorite TV shows. I don't know. Do people watch that? I think it's such a uplifting one, and it's really quite good. So quite good. Okay. So this is by transamination. Okay, and then glutamine is going to donate, and we're going to go through this. Okay. So these are the amino acids that we're going to be looking at. Some of them are actually just reverses of the breakdown. Okay. So the first is alanine. Okay. This is the direct synthetic pathway. So this is the anabolic pathway, and this is the breakdown for glycine, serine, and cysteine on this side. Okay, so this is the anabolic. So this is the catabolic. This is the anabolic side. Okay, so what's going to happen is we take glucose, and eventually it becomes 3-phosphoglycerate. Okay, and then it gets converted to 2-phosphoglycerate, and it goes to pyruvate. And then it magically becomes alanine by what kind of a process? Transamination, that's the word, okay? So it's relatively easy, okay? So that's what we want to do, okay? Good. All right. Now we have serine. We've actually seen there are a number of pathways for making serine. We already saw one, which we'll come back to, I think, in the next schematic, okay? So here, we're taking it, so we're going to make it from glucose by glycolysis. We make 3-phosphoglycerate, and effectively there's a dehydrogenase, so we get 3-phosphohydroxypyruvate. You don't really need to know all of these steps. 3-phospho-L-serine. There's a phosphatase that takes it off, and we end up with serine by that process, okay? All right? So serine is made from glucose, and it goes to serine through the glycolytic pathways, okay, on that. So where did the amino group come from on 3-phosphohydroxypyruvate? The amino group came from glutamate, okay? Notice this PLP, which is, yes, pyridoxal phosphate. Okay, don't answer all at once, okay? Maybe if you all whispered the answer, okay, I could hear what it was, okay, as a group, okay? 3-phosphoL-serine. Then there is another pathway, this is really the breakdown pathway, and effectively serine can be broken down to hydroxypyruvate, to glycerate, and all of these things. You don't need to know that, okay? Yeah.
So we don't need to know the in-between steps.
Yeah.
We just know that you get serine from 3-phosphoglycerate.
Right, and you do need to know the transamination step, okay?
Oh, just the transamination.
Yeah, so where's the amino group coming from, okay? I mean, effectively, you've seen these intermediates in glycolysis, okay? So the new thing is, how do I get the amino group on, okay? So the carbon structure is already done. You know it from glycolysis, okay? So you need to know the amino acid, the transamination, okay? Here's the other serine stuff, okay? So we saw this in the lecture on folate, that included folate, okay? And you remember that serine breakdown and threonine breakdown were the primary ways that you put carbon into it. It entered it as at what level of oxidation? Hydroxymethyl, right? Very good. Okay, because you're actually making. In this one, you're actually going to have when you have glycine cleavage enzyme and this, you're actually making N5-10 methylene tetrahydrofolate. You should always think of that as hydroxymethyl group. Okay. Now we only use it in one place, the hydroxymethyl form, the N5-N10 methylene. What reaction? Yeah. Dump to dethump, right. That's the only place that it's used. Most of it then gets actually oxidized, and we don't have it in the schematic, to, in essence, the formal form, because the formal one is the one that goes to making purines, all right? Do you remember? It's N5, well, it's either N5 formal, okay, or it's N5-N10 methanol, which is a shift space, and it will actually give you the same thing, okay? All right, so that's the way. So here's we can actually run in this direction. Remember, this is glycine cleavage enzyme in this direction, okay? In the other direction, it has a different name, but it's still the same process, okay? And it's called glycine synthase, okay? And why synthase and not synthetase? No ATP, right, very good, okay, all right. Don't let him answer all the questions, come on. You have to talk louder, okay? All right, so we're going to take CO2, ammonia, N5-N10 methylene tetrahydrofolate. We get folate, we take, and we use NADH, and we end up getting glycine. So this is the synthetic pathway for glycine, okay? In addition, okay, it can actually be a synthetic pathway for serine, because this reaction, serine hydroxymethyltransferase, is actually reversible. So we can make serine by that row and that way, okay? I wanted to add one other thing, and then threonine actually is broken down to glycine this way, and it's not reversible. What I wanted to look at is this D-amino acid oxidase, so it uses molecular oxygen. It's actually forms peroxide, so it's really not a mixed function oxidase, and we form glyoxylate. And glyoxylate gets converted to this dicarboxylic acid, oxalic acid, Which, if I taught it to you, I told you that the dicarboxylic acid names are, oh my, such good apple pie, and it's oxalic, malonic, succinic, glutaric, adipic, and pimelic, but you don't need to know apple and pie because you don't need to know it at this point, okay? You only needed to know for lysine synthesis, okay? Adipic and that, so that's how you can remember the dicarboxylic. The problem with oxalate is oxalate can actually precipitate out in your kidney and cause urinary stones, okay? And so it's oxalate. What would be, if you wanted to avoid foods that were high in oxalate, what would be you could think about? You may not know this, but it's important for you to know. Spinach is filled with oxalate. So for those who are eating spinach to look like Popeye, okay, and to in essence increase their blood, their iron, okay, when on that basis, you actually include it. So leafy greens, probably kale as well, okay, leafy greens have a lot of oxalate in them, okay, and so somebody who's doing oxalate stones. Oxalate is one form of stones. It's calcium oxalate that they form. The other ones can be, in essence, other forms of, there can be bile salt stones and everything. So one of the things, if somebody has and is passing kidney stones, they will want you to capture them and use a tea strainer to, in essence, urinate through so that you can select it, so that they can decide what it is, whether it's too much calcium or too much oxalate or whatever. So that's just one of the important things that you need to know. Okay, so that we don't need to know anything else. Okay. Now here we have cysteine. We've seen this already when we looked at the breakdown of methionine, okay? So the breakdown pathway for methionine is the synthetic pathway for cysteine, all right? So it's not like we've invented something else, okay? So this is really just a review. So here we have methionine, which is going to go to SAM, and then SAM is going to go to SA, and the methyl group is going to end up being put on homocysteine, okay? And that's what we're doing. What we're going to get is homocysteine when we take when we take the methyl group off. So in this situation, we're going to start with a molecule of serine, which is C3, and a molecule of homocysteine, which is C4, and we're going to combine them to make cystathionine, okay, which is this molecule, all right? And then what we're going to do is we're going to break this bond and then effectively get cysteine and alpha-ketobutyrate. Alpha-ketobutyrate goes to this pathway, which is, we know as what? Two names. Propionyl-CoA to succinate. Odd-chain fatty acids give you propionate. And what else? The vitamin M. Don't look for that on the boards, so the mini boards, all right? Because the vitamin M is my invention of way of remembering it, okay? The vitamin M ones, okay? So when they're dealing with it, we're going to get cysteine that way, okay? Don't even worry about this. We get sulfites and a whole bunch of other things which you don't really need. So this is the synthetic pathway for cysteine, okay? Notice that this enzyme, cystathionine synthase and cystathionase, are both PLP enzymes. If you have a deficiency of pyridoxine, okay, you can actually affect this. So what do you think would happen with a deficiency of pyridoxine? How would it present? Or what would you find in the blood or urine? If you were missing PLP, both of them, what would you find in the urine? Take a guess. Yeah. What? Say it again, louder. Homocysteine, yes, very good. Okay, it's a perfect answer. If you had said it louder, I would have been, I would have heard it the first time, okay? All right, so you don't have to be shy, okay? Homocysteine, okay, so that would be that way. And what would happen also, you would also find cystathionine. So you would have a cystinuria, homocystinuria, meaning urine, and a cystathionuria, okay, in both of those. And then you also get that, not both. It's a shortage of PLP that you get both. Okay, if you're missing this enzyme, you end up with homocystinuria. If you're missing this enzyme, okay, you're going to have cystathionuria, maybe a small amount of homocystinuria, okay, on that basis. This then becomes, cysteine becomes an essential amino acid in somebody who has that property, yeah.
You mentioned you created a system called the M vitamins.
Yeah,
What is that?
vitamin M.
That a little bit, vitamin M.
So it's valine, valine, right? Isoleucine, threonine, and methionine.
Why did you call it M vitamin? Oh, thank you, I guess.
V is valine,
Valine,
I is isoleucine, T is threonine,
and methionine.
nine is the
I'll
M.
never forget it. Thank you so much.
Okay, but thanks for asking, okay? The only stupid question is the one that isn't asked, okay? All right, so, all right. So, are any questions about that? So that's relatively straightforward. Okay, glutamate. Remember, glutamate is really structurally important for the way to make glutamate. There are a couple of ways to make it, okay? But this is the primary way. So what's going to happen is we're working in this direction, we're working in this direction, going this way. So we're going to start with a keto acid, alpha-ketoglutarate, C5. Remember, glutaric acid is oh my such good. O is 2, my is 3. O is oh my such succinic, and then glutaric acid. Okay, so alpha-ketoglutarate effectively is this. We're going to take NADP, H or NADH, and ammonia, and we're going to actually add it on by glutamic acid dehydrogenase. We know it from the urea cycle in this direction because it's one of the major ways to produce free ammonia, okay, and dealing with it that way, okay? And that's one of the reasons that we transaminate usually with alpha-ketoglutarate, all amino acids that transaminate to get to alpha-ketoglutarate, because alpha-ketoglutarate can go then directly in the liver to ammonia and alpha-ketoglutarate being regenerated. Glutamate can be that way. Is that clear to everybody? Okay. All right. So, so this is the major way that we're going to make it, and we put the amino group on. And it's not by a transamination. It's by a dehydrogenation, and it's a little more involved. Okay? All right. So, now how do we make glutamine? This is a very complicated process. Okay? It's one enzyme. Okay? All right. Yes? Say it up. Talk. You got to talk louder. Oh my, such good apple pie is the dicarboxylic acids. It has to start with 2 because you have to have more than one. So it's oxalic is 2, malonic is 3, succinic is 4, and glutaric acid is 5. You don't need the apple and pie. It's adipic acid and pimelic acid because you don't need it that way. All right? And we've done oxalic we haven't looked at, although we've looked at, when you're looking at, well, pretty much. But we did look at malonic acid, right? Do you remember what malonic acid had, what effect did the malonic acid have on what pathway? Can you name the pathway? Yeah, you're close there, but just malonic acid, not malonyl-CoA. You could think.
Oh, that's what I was thinking about.
Malonyl-CoA, okay? That's the building block. But malonic acid is there. It's a C3. Remember, where you have malonyl-CoA, when you're adding 2, you actually, CO2 is coming off, and then remember that drives the reaction for synthesis. So you're right that malonic acid is there. Malonic acid is an analog of what? A homolog, I'm sorry. Think about the Krebs cycle. Is there a dicarboxylic acid in the Krebs cycle? Yes. What? Fumarate it's a dicarboxylic acid, but it has a double bond. But just a plain ordinary one. Is there a plain ordinary one? Before fumarate? Before succinyl-CoA, after succinyl-CoA. Succinate, okay. Succinate is a 4-carbon, right? So if you you have cells undergoing TCA cycle, okay, and you put in malonic acid, it binds to succinic acid dehydrogenase, and since there aren't two hydrogens for it to take off, okay, it in essence is the perfect example of a competitive antagonist, a competitive inhibitor, okay? So we've seen malonic acid, okay? And what about, what was the next one? So, such succinic acid we got there, and then glutaric acid is alpha-ketoglutarate. That's so you know it's a C5. That'll help you. Remember, the amino acids, glutamate and glutamine, go to C5, alpha-ketoglutarate, and the C6s, histidine and arginine are the 6s, okay, that go in there, so the glutarate, okay? I'm sorry I didn't tell you that when I was teaching you carbohydrates. The it really helps you a little to think about the number of carbons and dealing with it. Did that answer your okay, good. All right. Thank you for interrupting me, okay, because if I want to make a point, it's very useful to do it, and we have reasonable amounts of time today, okay? All right. So here we have glutamine, okay? So here we start with glutamate, okay? And so this is the synthetic pathway. We take ammonia, ATP, and we have glutamine synthetase, and we make, we convert it, the ATP goes to ADP and PI. And so we make the amide nitrogen. Everybody's clear that this nitrogen is the amino nitrogen, and this one is the amide nitrogen. We have C double bond O, okay? C double bond O, NH2, okay? This one can't be protonated because the electrons that would be available for protonation are actually in here, in this carbonyl group, okay? So it doesn't take up a proton, okay? Glutamine synthetase is really important in the brain. There's a lot of glutamine in the brain. There's a lot of glutamate in the brain. Why? Because glutamate is what kind of a neurotransmitter? What type? Excitatory, right? None. Okay, excitatory, right? Okay, it's excitatory. We'll come back to that again, okay? So you're going to do it that way. So why in the brain would we be making glutamine? What does making glutamine get rid of? Ammonia. And so you're going to make glutamine in the brain and in other tissues, but in the brain where remember, ammonia isn't good. Remember, it's not a good thing to have. So you make glutamine, and now you let this go into the plasma. What's the benefit? One, it moves two amino groups, and two, it's neutral. Okay, because this one it can't be protonated, so effectively this is the negative charge is kind of canceled by the positive charge, so it's a neutral amino acid. So I don't change the blood pH by using glutamine, and I move two nitrogens to the liver. So what's going to happen? Okay, so what happens is in this situation is that the glutamine then is picked up in the liver, and then glutaminase removes the amino group, the ammonia, which we gave you, glutaminase and asparaginase, when I gave you the reactions that gave you free ammonia, okay? And then we get glutamate, and that's then worked on by glutamate dehydrogenase to free the nitrogen there, okay? So that's kind of what's happening to feed the free ammonia to effectively going to the synthesis of urea, okay? All right, everybody comfortable? Yeah.
So glutamate by itself, right? It's an excitatory.
Yeah, glutamine is not a neurotransmitter.
But when you have like the GABA is made from glutamate, right?
Right.
And then,
That's
so
what
then
you-
that
but
one's
you-
inhibitory.
What? Yeah, and that's inhibitory, yeah.
Okay.
So you get a balance between the two. Okay, we're going to see that, okay? All right, everybody kind of comfortable with that? Okay, good. All right. Proline. Proline is the amino acid that we really don't talk about too much unless you're in a collagen, talking about collagen. And then it's every third amino acid in collagen, and collagen is proline or hydroxyproline. Okay, hydroxyproline is made on the collagen by hydroxylation, by proline hydroxylase. Okay, so there is no code for it, so it's proline that's coded for. So Okay, so in this situation with proline, proline is if you wanted to count it, it has 5 carbons. Okay, it's going to be made from glutamate, which is 5 carbons, okay, which is the amino group, the glutamic acid, okay? And so what's going to happen is that this is the breakdown, this is the synthetic pathway over here. So what's going to happen is we're going to, when you see NADPH and NADP, you know that this reaction is number 1 is what kind of reaction? When do you see NADPH and plus H plus? In reduction. So this is a reduction, okay? And we're going to get some glutamic acid semialdehyde. The carboxyl, here's the carboxyl carbon. It's not being changed, but this carbon, okay, which is effectively becoming more reduced, right? It's going from a carboxyl group to an aldehyde. Everybody kind of comfortable with that? You're assuming NADPH, we get semialdehyde, okay? So, what happens, okay, this one, 1, 2, and 4 occur, I want to make sure, 1, 3, and 4 reaction. This reaction, and that reaction occur in the mito, and number 2 occurs in the cytoplasm, okay? This spontaneous cyclization of this, so effectively what's happening when this carbonyl interacts with that ammonia? What kind of a bond are you forming? He says a shift base. Everybody agrees? Yeah, that's what happens. That's how they interact, okay? Right? Everybody see it? So you're forming. Remember I said you're going to see shift bases in these lectures all over the place, okay? So what happens in this situation then is we get a shift space. Here it is. C double bond O, C double bond N, all right? And then what's happened is that's reduced because we want to get a hydrogen here, and we get proline. Relatively the same, okay? So the important thing is to know that you have to go through this process of making the glutamic acid semialdehyde, and then you make the proline, and there's no enzymatic process, it's spontaneous. Questions about that? Okay, yeah?
You mentioned that number 2 occurs in the cytosol.
Hmm?
Number 2 occurs in the cytosol.
Right.
And what else?
And 1, 3, and 4 occur in the mito.
And sorry?
So these are the breakdown pathways. So this is proline goes to FAD. In essence, you're actually oxidizing this to get the double bond in here, and then you're going to go in this direction, and now you're going to reduce this to you're going to oxidize it and produce NADH and effectively regenerate glutamate. So this side is synthesis, this side is that. So 1 is mito, 2 is cyto, 3 is breakdown, and it's in the mito, and 4 is in the mito as well, okay? Everybody? Okay. So here's ornithine. Ornithine is not coded for in proteins, okay? But and we normally make it from arginine, okay, by arginase, and we make ornithine that way. But there is another way, and you start with ornithine and alpha-ketoglutarate. Okay, so the synthetic pathway is going to be this way. So what we're going to do is take glutamic acid. That's going to be the source of the amino group. Okay, and this glutamic acid semialdehyde would be this where we got it the same way. Okay. So we would go from glutamate to this. So 1 molecule of glutamate goes to glutamic acid semialdehyde, and the other one goes to glutamate. Okay? So this semialdehyde is going to contribute the carbons, 1, 2, 3, 4, 5, and then this is going to contribute the amino group. So what we're going to do is what kind of a process? We've got an aldehyde up here and an amino group over here. So this process is a transamination, okay? Ornithine aminotransferase, okay? But you don't have to know it. It's a transamination. The amino group ends up in this position, okay? And the keto group ends up right here, okay? Yes? You have a question or spinning your pencil? Okay. You're spinning your pencil now. So it's contagious. It's like yawning, okay? It's contagious, okay? Everybody kind of comfortable with this? All right. Okay. All right. Okay. Never seen this before. Okay. All right. So how do I make arginine? Okay. So I make arginine going through this whole process, 1, 2, 3, 4, argininosuccinate, and you're going to end up making arginine, okay? Interestingly enough, this is a guanidо group. We're going to see it being transferred from arginine into another molecule when we're going to see a product in the next lecture, okay? So this is a guanidо group, and that's really important, okay? And we end up by cleaving it by arginase and make ornithine in this process, okay? So effectively we make arginine, and so it's only in youths and in disease when you don't have enough arginine being made, okay, that you would probably synthesize ornithine and do it that way, okay? All right. Okay. Aspartic acid and asparagine. Okay. How many carbons in aspartic acid? Choices 1, 2, 3, 4, or 5? 4. Okay. All right. That immediately tells you if it's going to interact to become glucose, you're going to think about what? What keto acid? The 4s go into the 4. What's the 4 in the Krebs cycle? Oxaloacetate, right, very good, okay, all right. I'm glad you're starting to, so I can hear you, okay, so it's, that's one of the things that my eyesight isn't going and neither is my hearing yet, okay. So we're going to start with oxaloacetate and it's going to be transaminated to make aspartate, so this is the synthetic pathway for aspartate. Notice transaminations always require PLP, okay? On that basis, okay? The same for ornithine, that transamination had PLP as well, okay? We get aspartic acid, all right. We're comfortable with that, okay? So now we're going to synthesize asparagine. Very hard process, okay? I hope you realize I'm kidding around, that it's not terribly hard. Okay, so what happens in this situation, this is worked on by asparagine synthetase, okay? Now here's a strange one, okay? Because normally when we're making an amide, normally it's not a transamination. Remember I said to you the two major nitrogen donors are aspartic acid, which we see in the urea cycle, and glutamine. And in this case, we're actually donating not the amino nitrogen, but effectively the amide nitrogen, okay? And it uses ATP, and that's asparagine synthetase, okay? All right. This enzyme has an interesting usage. It's actually used as a drug for treating cancers. Some cancers are asparagine is an essential amino acid. So what will happen is that I'm sorry, on this side, the asparaginase is the interesting one. So you give them asparaginase, and it breaks the asparagine that they normally have as an essential amino acid. It breaks it to aspartate, and in essence limits the development of the further development of the cancer. So it's one of those strange times when an amino acid, when a protein is actually used as a drug for treating cancer. All right, and part of the problem is that it actually will be taken up and function and work that way. So it's there. Yes,
So is
you got your hand up facing that way, so I
I'm
wasn't
so
sure.
sorry.
Okay,
I'm sorry, I apologize.
I wasn't sure if you were flagging
I'll
over
do that again.
that cab.
Is alanine also a nitrogen donor?
What?
Alanine.
No, it usually isn't. Okay, it can, but it usually transaminates to proline and whatever.
So it doesn't
it
transfer
can-
nitrogen?
It can be. Most of the time, the nitrogen donor is glutamate. Okay, but alanine can, and there are proline. I'm sorry, there are alanine keto acid transaminases. Okay.
So for the nitrogen donors, just remember aspartate and glutamine? Okay.
Yeah. No, not for nitrogen donors. For nitrogen donors to large molecules. Okay. So, for instance, in purines, which one is a donor to one of the rings? Oh, which are the primary donors to the two rings of purines?
Purines,
Purines is the big molecule.
so it would be the glutamine?
Glutamine, the two nitrogens on the bottom. Okay.
And aspartate for pyrimidine.
Did for the nitrogen on the top, okay? And so that's what I'm saying is you're making a large molecule, and those are the donors, okay?
Oh,
Now the other nitrogen is also coming from glycine, okay?
okay.
But not in the same way. The glycine nitrogens are, the other part of glycine is actually there, okay?
Thank you so much.
Does that help? Okay, yeah, okay. You know, sometimes talking about it and listening, it helps you to kind of formulate theories and or correct errors, and so I think that's useful. That's very useful. Okay, so asparagine. This was asparaginase we gave you as another enzyme that forms ammonia that way. Okay. Any problems with this? Okay. Okay. So. This is going to be relatively easy because we've looked at this same pathway already, and we talked about all the stuff in here, homogenesic acid, alkaptonuria, and we've done all of that. So this is really looking at tyrosine. So tyrosine is made, it's not an essential amino acid, it's made from phenylalanine, okay, by phenylalanine hydroxylase, okay. What's the major cofactor? For what? BH4. Biopterin. Biopterin, yeah, that's the B part, okay? So tetrahydrobiopterin, okay? We're going to see it again in another place, okay, on that basis, okay? Here. So tetrahydrobiopterin, and PKU is the deficiency, okay? So this is just a review of how do I make this and this, okay? Everybody comfortable? Any questions about that? Okay. Yes? It's not. It's something that we synthesize. Okay, it's a pteridine of sorts. Okay, and it's not considered a vitamin. It's one of those cofactors that isn't a vitamin. Okay, all right. And we're done. Okay. So, are there any questions? This is a relatively straight one because a lot of times they're just very one step. You know, you got to know glutamine synthase, you got to know asparagine synthase, and set it up that way. They're relatively straightforward. So now, some of you, how are you doing? A lot of people thought that you would have to learn all these pathways and stuff, and the answer is you just have to have a general idea of where they're coming from and what's happening. Okay, all right, and whether they're essential or not, essential, and dealing with it that way. Okay, good. So why don't we take like a 5-minute break, and is that okay? And then we'll maybe make it a 10-minute